Hello, my name is Joshua Mugabe. I welcome you to Chromosomes. Our lesson objectives today are: we shall look at chromosomes, we shall look at plasmids, we shall look at chromosome numbers, haploid versus diploid, autonomous versus heterosomes, caliograms, and autoradiography. You are welcome. Do not forget to subscribe. Organization of DNA. We say that DNA is double-stranded molecule that function as the genetic material. DNA is double-stranded molecule that functions as the genetic material of a cell. So it has two strands. If you look at it, it has two parallel strands running parallel to each other, joined by bases in between. Those are the ones that we are referring to as a double-stranded molecule, and it functions as the genetic material of the cell. It contains the genetic information. It is composed of the nucleotides made up of 4 bases. And those bases that we have adenine, thymine, guanine, and cytosine. So that is the DNA. It is composed of nucleotides which are made up of 4 distinct bases: adenine, thymine, guanine, and cytosine. And then the order of the bases form gene sequences. The order of these bases form gene sequences that codes for the specific proteins. So when these bases are arranged in the strand, they form what we call a gene sequence. And that gene sequence is the one that codes for the specific proteins, as we shall see later on. A single double-stranded DNA molecule is called a chromosome. A single double-stranded DNA is called a chromosome. You will see that you will find sometime a structure which is like that, and we call that one a chromosome. That chromosome, when you look at it, it looks as if it is a single-stranded, but it is double-stranded, though it is appearing single, but it is double-stranded. So when it is like that one, we refer to it as a chromosome. A single chromosome may contain thousands of unique genes on either strand. A single chromosome may contain thousands of unique genes on either strand. So we are saying that a gene is double-stranded, meaning that you have a chromosome which is like that. It has 2 strands, and these 2 strands are connected by a pair of bases. So that whole of this one, these 2 strands, are what we are referring to as a single gene. It's the one that we call a single chromosome. And this single chromosome contains thousands of unique genes on either strand, So at every point of the chromosome, at every point of this chromosome, they are what we refer to as the genes. It may have very many genes on each strand, and each strand has got unique and different genes. So there might be very many genes on every strand. It is a single chromosome, but made up of double strands. And that single chromosome contains thousands of unique genes on either strand. Chromosome structure differ between prokaryotic and eukaryotic organisms. So we have organisms that we call prokaryotic. Prokaryotic organisms, these are organisms that don't have membrane-bound organelles. And the eukaryotic organism, these are organisms that have got membrane-bound organelles, or they have a nucleus. So their chromosome structure is different from those ones of prokaryotic to those ones of eukaryotic organisms. If you look at their chromosome structures, they are different. They differ. Chromosomes are circular in prokaryotic. Chromosomes are circular in prokaryotics, while in eukaryotes, they are linear. In eukaryotes, they are linear. So in prokaryote, you find that the chromosomes, they are circular. That is in prokaryotes. And then in eukaryotes, you'll find that their chromosomes are linear. They're just a straight line. Prokaryotes, these are organisms or cells that don't have a nucleus, or they don't have a membrane-bound organelles. Whereas eukaryotes, these are organisms that have a nucleus, and also they have cell membrane-bound organelles. Prokaryotic chromosomes. Let's look at the prokaryotic chromosomes. Prokaryotic chromosomes do not possess membrane-bound nucleus. They don't possess membrane-bound nucleus. For example, if you look at our nucleus here, this is a circular DNA, which we call the gynophore, and it is for the prokaryotes. When you look at it, it is circular, but it has no membrane-bound nucleus around it. So it is just floating in the cytoplasm. No nucleus at all surrounding it. The DNA is contained in a region called nucleoid. So in a region where this DNA is, it is referred to as a nucleoid. So this DNA is not in a membrane-bound nucleus, but it is just a region in the cytoplasm, and that region where we find it, we call that region a nucleoid. Prokaryotes have only a single chromosome. They have only a single chromosome, and that chromosome we refer to it as gynophore. So they have only one single chromosome, and that chromosome is what we are calling the gynophore. And when you look at it, you can see that this chromosome is circular. It is circular in structure. That is the DNA of, or the chromosome of a prokaryote. It is a single circular chromosome. The DNA is circular, that one we have already seen, and naked, meaning that it is not in a nucleus. It is not bound to proteins. It has no histamine. It is not bound on proteins, and it is circular, and it is naked. It does not have a nucleus, but it is in a place which we call a nucleoid. That is for the prokaryotes. The genome is more compact. The genome is more compact, and usually no interons. There are no interons. So its genome, the totality collection of all the genes, is compact. It is compacted together. Some eukaryotic organisms also contain or possess circular DNA. We have also some organelles which are prokaryotes. These are organisms that have cell-bound or membrane-bound organelles. For example, there are some of them which possess circular DNA. And those examples, we have the chloroplast, and then mitochondria. These are eukaryotic organelles, but they also possess circular DNA. This one now is an indicator that these organisms might be having a prokaryotic origin. They might be having a prokaryotic origin. Why? Because they have a circular DNA in them, and that is chloroplasts and mitochondria. Prokaryotes have one chromosome. Prokaryotes have one chromosome, and this one consists of a circular DNA structure. We have already seen it here. This structure is their DNA. So they have only a circular DNA molecule. And this circular DNA molecule is the one that we call the gonophore. Let's look at the plasmids. Now, prokaryotes may contain an extra DNA. We have seen that they contain a single circular DNA. But again, they may also contain an extra DNA molecule in addition to the gonophore. Gonophore is the major DNA molecule of prokaryotes, which is circular. But again, they may contain another extra DNA molecule in addition to the gonophore. So here we have our gonophore. This one is our gonophore, which is circular DNA or chromosome. And then they also have another extra DNA, which we are calling the plasmid, or what we refer to as autonomous DNA molecule. These molecules are called plasmid and are capable of autonomous function. These other chromosome or DNA structures, we call them plasmids, and they are capable of autonomous function. They are capable of doing separate work, separate function. They are capable of doing independent functions. Those are the extra DNA, which we are referring to as the plasmid. But the real DNA molecule is the one that we call the gonophore. Prokaryotes may exchange these plasmids by bacterial conjugation. They may exchange these plasmids by bacterial conjugation via the sex pili. So the plasmid can be exchanged from one prokaryotic cell to another prokaryotic cell through the sex pili. And that process is conjugation. It's referred to as conjugation. Conjugation is where the prokaryotes exchange the plasmids via the sex pili. It's what we refer to as conjugation. Eukaryotic cells, these ones do not contain plasmids. They don't have plasmids unless they are genetically modified. So eukaryotic cells, these are cells that contain organelles that are bound with membrane, or membrane-bound organelles are found in cells which are called eukaryotic cells. And for them, their DNA is enclosed in a nucleus. As we have also talked about prokaryotic cells, they don't have a nucleus, or they don't have, their chromosomes are not bound in a nucleus, and they don't have membrane-bound organelles. Some prokaryotes also have plasmids. Some prokaryotes also have plasmids, but eukaryotes do not. Eukaryotes do not. Eukaryotic cells don't have plasmids. Eukaryotic cells, they don't have plasmids. But prokaryotes are the ones that have got plasmids. Though we have seen that some organelles in eukaryotic cells, they might be having plasmids, for example, chloroplasts and mitochondria, but those are organelles. But the eukaryotic cells themselves, they do not have plasmids. Eukaryotic chromosome. Eukaryotic chromosomes. Eukaryotic DNA is stored in a membrane-bound nucleus. The eukaryotic DNA is stored in a membrane-bound nucleus. So for the eukaryotic cells, they have a nucleus, and inside the nucleus, that's where we find our chromosome. So the chromosomes are bound by membrane called the nucleus. Those are eukaryotic cell or eukaryotic DNA. The DNA is bound on histone proteins to form what we call chromatins. So because the chromosome is a very long structure, they have proteins which are called histones. These proteins, the histones, they are in particular points, and in those particular points, what happens is that the chromosome comes and bounds around the histones. They bound around the histone, and when they bound, the chromosome bounds around the histone. This one makes it to become shorter, and when it becomes shorter, now it forms what we call the chromatin. They have become what we call the chromatin because the DNA is bound on the histone protein to form a chromatid. For example, here we are seeing a linear DNA condensed. The linear DNA condensed. When you look at its length, you will see it is shorter because it is rotating or bound around the histone proteins. So that makes it to become condensed or to become shorter. and then now we can refer to it as chromatin. Now, we are saying the genome is less compact. It has many introns. The genome is less compact. That is for the DNA or the chromosome for eukaryotes. The genome, the total genes or total number of genes, they are less compact because they have got a lot of introns. Introns, these are some of the chromosome parts which are not bound on the histone. There are chromosome parts which are not bound on the histone, are the ones that we are referring to as the introns. So they have many introns. Therefore, the genome now appears as if it is less compact. The chromosomes will exist as uncondensed chromatin, except during cell division, to form what we call visible chromosome. So the chromosome, always they appear uncondensed. It is just like a single thread, very long thread. That one is what we call uncondensed chromatin. And when do they become condensed? They become condensed when they rotate around the histone proteins. We have looked at the histone proteins. The histone protein, what happens is that the chromosome will come and rotate around the histone proteins, and then it becomes shorter in length. So now it has become condensed. When it is bound around the histone protein, it becomes condensed. But when it is not undergoing cell division, if it is not undergoing cell division, then it will appear to be condensed. It will appear to be condensed, just like a single long thread. It is uncondensed. There it is uncondensed chromosome. But when it is undergoing cell division, then it becomes condensed. It is bounding itself on the histone protein and it becomes condensed. Therefore, there it becomes like a chromatin. So, eukaryotic chromosomes are linear DNA molecules associated with histone proteins. Eukaryotic chromosomes, they are linear DNA molecules associated with the histone. They are associated with histone proteins. Histone proteins, we have talked about the histone proteins. This is where the linear chromosome is bound. And we are saying that eukaryotic chromosomes are linear DNA. They are just made up of a single structure, single line molecule, but they are associated with the histone protein. At a particular point, you will find histone proteins, and on those histone protein, it's where the chromosome comes and bind itself. The chromosome rotates around the histone protein. But in the real normal structure, the eukaryotic chromosomes are linear in structure. Chromosome composition. And okay. We are looking at chromosome composition. Now, eukaryote organisms, they have multiple, that is more than one chromosomes, and each one carrying many different genes. Those are eukaryotic organisms. They have more than one chromosomes, and each of those chromosomes is carrying very many genes. These genes, they are located at specific gene loci. The genes are located at specific gene loci. Now, if you look at the human chromosome here, for example, we have different chromosomes here. There is this chromosome number 3, chromosome number 11, chromosome number 15. Those are our chromosomes. And we are saying that eukaryotic organisms, they have multiple chromosomes, very many chromosomes. And these chromosomes, they contain different genes. Now, what we are seeing, these ones that are running horizontal, those ones are loci, where we find our genes. Those are our genes. And where we find the genes, those points or that location is what we are referring to as a loci or loci or locus. So the genes are located at specific gene loci or loci. Those are the position. So we have the chromosome represented by a number. This is chromosome 3 with different genes, and those genes are located at specific gene loci. Different chromosomes will have distinctive sizes. They have distinctive sizes and other features like the arm length and the banding pattern. So, if you look at the different chromosomes, what you are going to see is that they have distinctive sizes. For example, if you look here, we have chromosome 3 is the longest here, chromosome 11 medium, and chromosome 15 is the shortest of the 3. Now, those are distinctive sizes. And they also have other features like the arm length. For example, if you look at this one, it has 2 arms. This first arm from here to here, that is the P arm. This one from here to here, that is also the P arm. And this one from here to here, that one is also a P arm. Now from here downwards, that is a Q arm. This one is a Q arm, and this one is a Q arm. So they also have arms of different length. As you are seeing, all of them, they have different length. Or if you look at the P, they have different length. Now, on every arm, there is what we are referring to as the banding patterns. If you look, this is a pattern, this is a banding pattern, banding pattern, banding pattern on the P arm. And this is a banding pattern, banding pattern, banding pattern. If you look at those ones, those are banding patterns. They are different. They have got different banding patterns on different arms. Gene loci or loci, the gene loci, can be identified according to the chromosome number, arm, and banding region. So when we are finding the gene location, what we are calling gene loci, that one can be identified according to the chromosome number. You can give the chromosome number, you give the arm, and then you give the binding region. For example, chromosome 7. Here we can say, for example, chromosome 3. This is now the chromosome number. The arm, we are looking at the P arm. This one up here is the P arm. And then you are looking at the binding region 31.3. 31.3 region is somewhere here. That is around 31.2. So that's how we can locate the gene loci or gene location. We give the chromosome number, the arm, and the binding region. Let's look at the chromosome numbers. The chromosome number is a characteristic feature of any given species. The chromosome number or the number of chromosome is the characteristic feature of any given species. So here what we are saying is that every species or any given particular species can be identified by that characteristic feature, and that characteristic feature is the number of chromosomes it has. So that's why we are saying the number of chromosomes is a characteristic feature of a given species. Now let us look at the different species here. We have a horse, we have a donkey, and we have a mule. These are different species. How can you identify them? You use their characteristic feature, and the characteristic feature is going to be the number of their chromosomes. If you look at the horse has 64 chromosomes. If you look at the donkey has 62 chromosomes. And the mule has 63 chromosomes. So those are their characteristic feature for those given species. That a species of horses, it has 64 chromosomes. Species of donkeys, it has 62 chromosomes. And the species of mule, it has 63 chromosomes. Now, different species have different chromosome numbers and gene distribution. If you look at the different species, they have different chromosome numbers and gene distribution. It's what we have been talking about. The horse has 64 chromosomes. The donkey has 62 chromosomes. And the mule has 63 chromosomes. So they have different chromosome numbers. That means that they are of different species. Members of different species cannot sexually reproduce, as the genetic content of their sex cells is not complementary. That is, they will have incompatible gametes. For example, we have here a species of the horse. We have another species of the donkey. These two different species cannot sexually reproduce. A donkey and a horse cannot sexually reproduce. Why? Because their genetic content in their sex cells is not complementary. So they have incompatible gametes. Though here we can see that a horse and a donkey can sexually reproduce and give a mule. But a mule is going to be infertile or sterile. It cannot produce or it cannot give a mule, and a mule cannot sexually produce another mule. So here it's what we are referring to as that members of different species cannot sexually reproduce, as their genetic content of their sex cells will not be complementary. Hybrids, we have hybrids. Hybrid species, that is the mule. A mule is a hybrid species. Can sometimes occur, but are always infertile. A mule is coming from a horse breeding with a donkey. But the mule now becomes a hybrid. And this hybrid, the mule, is infertile. It cannot produce a viable offspring because it is coming from two species which are incompatible or which are not complementary. Their sex cell component, genetic composition is not complementary. Haploid versus diploid. In sexually reproducing species, offsprings will inherit genetic information, that is the chromosome, from both parents. That is, in sexually reproducing species, offsprings, they inherit genetic information or chromosomes that are coming from both parents. Some chromosomes will come from the mother and other chromosomes will come from the father, maternal and paternal. Two copies of chromosomes of each chromosome. They have two copies of each chromosome. And that is what we call a diploid, 2N. For example, here, let's look here. This is a diploid, two copies. We have this set of chromosomes here. And you can see it is made up of two copies, meaning that one copy came from maternal and then another copy came from paternal. So the same applies also here. This is another copy of chromosome. One copy came from the maternal and another copy came from paternal. The same applies here. These are two chromosomes, so we refer to them as diploid. One came from maternal and then the other one also came from paternal. So that's what we are saying. Two copies of each chromosome. Two copies of each chromosome. This is chromosome number, can be number 3. This one can be chromosome 21. This one can be chromosome 13. But all of them, they are in two copies. One copy comes from the father and another copy comes from the mother. While all body cells will have paired chromosomes, the sex cells, those are the gametes, must only have one of each pair. So here we are looking at the body cells. Body cells, these are the cells that are not concerned with the sex. They are the ones that we call body cells. Sometimes we refer to them as somatic cells. They are the ones that we refer to as body cells. The body cells, for them, they have paired chromosomes. They will have what we are calling paired chromosome, or what we call the diploid. They have a chromosome. One came from the mother and another one came from the father. That is one set of chromosomes, and we call it a diploid. Then they have another chromosome number, maybe 13. This is chromosome 13 and this one is chromosome 13. But they are in pair. One pair came from the mother and another pair came from the father. Another one can be chromosome 17. This is chromosome 17 and this is chromosome 17. But they are existing in a pair. There is one that came from the mother and another one came from the father. So these ones are the ones that we are calling diploid. And these ones, we find them in body cells. Body cells are the ones that have got paired chromosomes. So this one and this one, they are paired. This one and this one, they are paired. This one and this one, those are paired chromosomes. Now, when it comes now to the sex chromosomes, the sex chromosome, these are the gametes. Sex chromosomes, they form the gametes. And these ones only have one pair of each chromosome. So if we have chromosome 13, chromosome 17, chromosome maybe 20, you will see that they have only one of the pair. This one might have come from the father or the mother. This one might have come from the mother or the father. This one might have come from the father or the mother. But they should be having only one set, one pair, one of the pair, each pair. So that one is what we call half the number of chromosomes. And it is what we refer to as haploid. They have only one copy of the diploid. So now we call it haploid because it is half of the diploid. Diploid, there are two copies. Haploid, it is one copy. One copy of each chromosome is referred to as haploid. And when we are writing it, we always write N to represent haploid. These ones now are haploid. It is only one copy of each chromosome is referred to as haploid. But if there are two copies of each chromosome, then that one is now what we refer to as diploid. Two copies of each chromosome. Those ones are diploid. But if it becomes one copy of each chromosome, one copy, now those ones we refer to them as haploid. Organisms that only reproduce asexually are haploid. So those organisms that only reproduce asexually, they are haploid. Those are organisms that do not use gametes for their sexual reproduction. Therefore, we refer to them as asexual or asexually. They are haploid, meaning that they have one copy of each chromosome. Diploid nuclei have pairs of chromosomes, while haploid nuclei have one chromosome of each pair. So what we are saying here is that when we have diploid nuclei, these ones, they have pair of chromosomes, while the haploid nuclei, they have one chromosome of each pair. So here, this is diploid, and we have pairs of chromosomes. This is a pair. Here we have a pair, and here we have another pair. Those are pairs of chromosomes. These ones are found in diploid nuclei. But when it comes to haploid nuclei, they have one chromosome of each pair. This is one chromosome of that pair. This is one chromosome of a pair, and this one is also one chromosome. Please do not forget to subscribe. Homologous chromosomes. Homologous chromosomes. Homologous chromosomes are the paired maternal and paternal chromosomes found in diploid organisms. So what we call homologous chromosomes, these are chromosomes that are paired, whereby there is one which is maternal and the other one is paternal. And these chromosomes, we find them in diploid organisms. So you will find that we shall have chromosomes pairing up. And when they pair up, now they become homologous. They become homologous. But one chromosome comes from the maternal and another chromosome comes from the paternal. So we have a maternal chromosome, at the same time we have a paternal chromosome. So when they pair up, they form what we call a homologous chromosome. Homologous chromosome. Homologous chromosomes will have one, identical structure features. They have identical structure features, that is, the size, the band pattern, and others. So if we look at this homologous pair, you will see that they have the same size. They have the same size. And also when you look at the patterns, band patterns, the band patterns are the same. So that's what we are saying, that homologous chromosomes, they have identical structures. They have identical structure or structural features that are identical, like the size and the band pattern. They have the same genes at the same loci. They have got the same genes at the same loci position. So when you look at the different loci, for example, if you look at the beginning here, you will see that this gene is the same as this gene here. And they are at the same locus on different chromosomes. If you look at this one here, it is the same as this one. And they are positioned at the same locus, though on different chromosomes. So we are saying that homologous chromosomes, they have genes at the same loci position. Because the paired chromosomes come from different origin, that is maternal versus paternal, the alleles may be different. So we are looking at these paired chromosomes. These paired chromosomes, they are coming from different origins. One chromosome comes from the maternal, and then the other chromosome comes from the paternal. So they have genes located at a specific loci. If we look at that gene there, this gene here, and we look at that gene there, it is the same gene, but they might be different alleles. Those alleles may be different because they are coming from different origin. This one here is a maternal chromosome, and this one is a paternal chromosome, but they are paired chromosomes, which we call homologous chromosomes, meaning that genes are located at the same loci or locus. But those genes that are located at the same point, it is the same gene, but they might be different alleles. So homologous chromosomes carry the same sequence. They carry the same sequence of genes, but not necessarily the same alleles. They may carry the same sequence of genes, but not the same alleles. This gene here is the same as this gene. If you look at this gene here, it's the same as this gene. If you look at this third gene, it's the same as this gene. So those genes at the same position or loci, they are the same genes, but they may be of different alleles. They may be of the same allele or different alleles. Autosomes versus heterosomes. Not all chromosome pairs in diploid cells are homologous. Not all chromosome pairs in diploid cells are homologous. Sex determination requires distinct chromosome. When we are carrying out sex determination, it requires distinct chromosome. Now, what are we saying? For example, if we look at our diagram, you will see that we have the X chromosome and we have the Y chromosome. If you look at them, they are not homologous. They are not of the same size, and I don't think the genes are the same. So if you look at them there, now that one will help us to understand that those are the ones that we use when we are identifying the sex. The Y chromosome is very different from the X chromosome. Though they are in diploid cell, they are diploid cells, and they are a pair, but they are not homologous. Humans, they have 23 pairs of chromosomes. And if it is diploid total, it comes to 46 chromosomes. That is a diploid. 23 pairs. 22 of these pairs are homologous autosomes. 22 of them, 22 pairs, are homologous autosome. Homologous means that each pair is the same. Autosome means that these are pairs which are not concerned with sex determination. The 23rd pair are the sex chromosome. The 23rd pair, those are the ones that we call the sex chromosomes. And we refer to them as heterosomes. Heterosomes. Hetero means different. So these are the sex chromosomes. So when you look at our X chromosome and you look at our Y chromosome, you can see that they are heterosome, meaning that they are not homologous. Therefore, that one is an indicator that those ones are sex chromosome. And that one is the 23rd pair of the chromosomes. The sex chromosomes are different in human males. For example, human males, they have X and Y, but they are the same, or what we call homologous, in the human females. Human females, they have XX. So you will find that for the human female, the sex chromosomes are homologous. They are the same in size and location of the genes at the same locus. But when it comes to human males, the sex chromosomes are different, meaning that they are not homologous. One is X, another one is Y. So they become different. Sex is determined by sex chromosomes. And autosomes are chromosomes that do not determine sex. Sex is determined by sex chromosomes, whereas autosomes are the chromosomes that do not determine sex. We have seen how we can easily identify sex chromosomes in males. We have X and Y, which are not homologous. Now, it might be a little bit difficult for the female sex chromosome, identifying the sex chromosomes of the female, because they are homologous. That is X and X. But sex is determined by sex chromosomes. Sex determination. Females, we have already seen, they have two X chromosomes. So they have X and X. And we have seen that they only pass X to their offsprings. The males, they have X and Y chromosomes. So they have XY. That means that they can pass either X or Y to their offsprings. The father is the one that determines the sex of the offspring. If X is passed on, then we shall get a female offspring. X from the father and X from the mother. If Y is passed on, then the child will be a male. X from the mother and Y from the father. We have the mother here. A mother is XX. And we have the father. The father is XY. So the egg, which is X, and the sperm, which is also X, they will give a girl XX. The egg, which is X, and the sperm, which is Y, we shall get a boy who is XY. Karyotypes. The chromosome profile of an individual organism can be represented as a karyogram. That is, the chromosome profile of an individual organism can be represented as a karyogram. The homologous pairs are arranged by length. Homologous pairs are arranged by length. Let's go to our karyogram. Chromosome pair 1, you can see that homologous chromosomes, they have the same size. If you go looking at them, all of them, they have the same size. Those are homologous chromosomes or homologous pairs. Karyogram displays chromosomes from cells that have been arrested during mitosis. And these ones are condensed DNA. So our karyogram displays chromosomes from cells that have been arrested during the mitosis process. That is the division of the cells, which results in the production of identical daughter cells. So that's where we get our karyogram from. And you will see that all homologous chromosomes are of the same size with the same binding pattern. Karyograms are generally used to identify the sex or to identify chromosome abnormalities, that is, aneuploidy. So if you have your karyogram, this karyogram is going to help you to identify the sex. It can help us to identify chromosome abnormality. For example, aneuploid. A karyogram shows the chromosome of an organism in homologous pairs. of decreasing length. So we shall see our chromosomes arranged in homologous pairs, and they will be of the same size, and the size will go on decreasing, starting with the long length to the shortest. That is how the karyogram is arranged. Karyogram examples. Karyogram is used to deduce sex and diagonalize Down syndrome. The karyogram is used to deduce sex and diagnose Down syndrome. Autoradiography. While condensed chromosomes are visible under microscope, DNA is too compacted to give reliable indication of length. Now we know that chromosomes, when they are going to undergo cell division, they become condensed, and therefore they become visible under a microscope. But when they are condensed, it means that the DNA is too compacted. And if it is compacted, it becomes difficult to rely on it to indicate the length of the chromosome. So John Keynes utilized a technique of autoradiography to visualize the chromosome whereas uncondensed. He used the autoradiography and he visualized the chromosome while they were uncondensed. Keynes technique for measuring the length of DNA molecule by autoradiography. They used a radioactive thiamide, which was incorporated into the DNA. Then the chromosomes were treated with silver bromide. The radiations, these ones converted silver ions into insoluble grains.
What I had been doing in Australia was looking at the multiplication of vaccinia virus by labeling the cell with tritiethymidine, which is a specific label for DNA, and therefore would label vaccinia virus, which is known to be a DNA virus. And it was a result of that, and looking at cells infected with it, that I discovered that each virus particle sets up its own factory. Well, when I went to Cold Spring Harbor, as I said, I came up with this fantastically complicated and ludicrous experiment. And then when that was shot down in flames by Bob Edgar, I had to think, and I thought, this is ridiculous. Here am I, Al's lab has worked out how to isolate DNA molecules without breaking them and how to handle them, and they had reached the conclusion that each phage particle contains a single molecule, and this is a T-even DNA phage. And I know about autoradiography, which is labeling with tritiethymidine and seeing the location of the label. why don't I see if I can measure the length of these molecules using this technique? And did some little calculations and decided it should be possible. It would take 2 months under film. So there were a limited number of experiments I could do. So that's what I did at Cold Spring Harbor. In the process, discovered one rather bizarre feature of doing experiments. You do an experiment that's going to last 2 months, and you tuck it away in a little light tight box. And the moment you've tucked it away, you realize you could have done a better experiment. So you now do a better version of that experiment and tuck that away in another light tight box. Consequence of that is you lost interest in the first light tight box. But now you think, well, gee whiz, the second was clever, but it'd be even cleverer if I did it in the following way. So the second light tight box is going to cease to be interesting. So you have all these boxes that you're not interested in, but you do develop the film when you get to the end, because you think, well, I might as well anyway. It's no trouble developing film. And so it is all these uninteresting experiments that you rely on in the end to get a result. So that's what I did at Cold Spring Harbor. And then we went back to Australia and stayed there for 2 years while I continued to do experiments on the length of DNA and the way it is duplicated and so on and so forth, looking at bacterial viruses and bacteria.
The preparation, he treated the cells, extracted the DNA, fixed it on the slide, immersed in silver bromide, and in the process, radiation activity nearby, silver ions, the ions were reduced to metals, soluble ions were removed, and the slide was exposed to the film, and then it was autoradiographed. Autoradiograph application. In addition to being used to identify DNA length, autoradiography has other useful applications. If radioactive uracil is used instead of thiamine, regions of active transcription can be identified. We can identify the regions of active transcription if we use radioactive uracil instead of thiamine. For DNA, tritiated thiamine is used. For RNA, tritiated uracil is used. More silver grains equals to more transcription. Now here, when you look at the grains of silver that were turned into metal, you can see that those black regions are the ones with silver. So if you have less black grains, then that means it is low rate of transcription. If you look down, here you have very many grains of silver. That one means that we have a high rate of transcription. So it can give you a position where you can see a lot of transcription on the DNA where it is taking place, and where you find less black dots. That means there we have low transcription taking place. Hello, my friends. Thank you very much for participating in this tutorial. We have looked at the chromosomes. We have looked at the plasmids. We have looked at the chromosome numbers, haploid versus diploid, autosome versus heterosomes, kardiograms, and autoradiography. Thank you for participating. Please do not forget to subscribe. Thank you.